Good morning, students. Myself, Dr. Ragasudha, Head and Assistant Professor, Department of Microbiology, Government City College, is here to talk on fermentor or bioreactor. Overview. Introduction. Design of fermentor, types of fermenters, types of fermentation, stages of fermentation process. Introduction. The term fermentation has been derived from the Latin verb fervor, which means to boil. This name was given on the basis of the boiling appearance of the action of yeast on extracts of fruits or malt grains during production of alcoholic beverages. The boiling appearance has been due to the production of CO2 bubbles caused by the anaerobic catabolism of sugars present in the extract. However, fermentation is interpreted differently by biochemists and industrial microbiologists. It can be defined as a metabolic process in which raw materials such as sugar or carbohydrates are converted into acids, gases, and alcohols. Microorganisms such as yeast and bacteria play a central role in the fermentation process. To an industrial microbiologist, fermentation means any process for the production of a product by the mass culture of microorganisms, and according to them, fermentation is defined as a process by which large quantities of cells are grown in a fermenter under controlled aerobic or anaerobic conditions which aims optimizing the growth of the organisms or production of a target useful product. Fermentation process requires a fermenter, substrate, inoculum and optimum conditions. The equipment used to carry out fermentation is called fermenter. Fermenter can be defined as an equipment or a vessel in which fermentation process is carried out. A fermenter is a complex machine. It is used to create optimum conditions that are necessary for the fermentation process. The main function of a fermenter is to provide a suitable environment in which an organism can efficiently produce a target product that may be cell biomass, a biometabolite, or bioconversion product. Therefore, it is a system within which substrate of very low value is employed by living organisms or enzymes to produce a product of greater value. Uses of fermenters. It provides to control environment for optimum biomass or product yield. It provides an aseptic fermentation condition for more reliability. It provides adequate or proper mixing and aeration for the growth and production without damaging the microbial cells. It has a system to monitor and regulate pH and control temperature of the fermentation broth. It facilitates sampling of the product. It facilitates easy harvesting of the product and the enzymes too. Let us see the design of a fermenter. Fermenter should be relatively large enough to allow creation of fermenter bacteria. The materials used in the manufacture of a fermenter should be strong enough to tolerate the pressures of fermentation media and gases produced. The material used for designing the fermenter should be non-corrosive. It should not contribute any toxic ions or substances to the fermentation media because these toxic ions can harm the growth of the microbial culture. It should have an inlet for oxygen and outlet for carbon dioxide in case of aerobic microbes are used for the fermentation process. In addition to that, it should be completely sealed from outside environment to avoid contamination. It should be easy for sterilization. It should provide a facility to add anti-foaming agents as well as temperature controlling system. It should have a pH detecting system, an outlet for withdrawal of the medium for determination of pH as well as inlet for addition of acids or alkali to control the pH. It is important to maintain pH at optimum levels for the growth of cultures. The most important thing is that fermenter should provide aseptic means of withdrawal of fermented product and introduction of culture samples. The fermenter should facilitate stirring of media so that inoculum and fermentation media mixes properly, and it also ensures that the gases required for growth of microbial culture are available to all the microbes. Therefore, a fermenter is basically a closed system vessel which has an adequate provision for temperature control, agitation, aeration, pH control, and drain or outflow vent to remove waste biomass of culture microorganisms with their products. The main parts of fermenters are agitation, which is used for mixing of cells and medium, aeration for aerobic fermentation, temperature control, pH, pressure, nutrient feeding, etc., sterilization and maintenance of sterility, withdrawal of cells or media. So this is the skeleton of a fermenter, a design of fermenter, wherein you can see it is a big vessel usually made up of stainless steel vessel, and at the topmost end you can see an anti-foaming agent inlet for anti-foaming agent addition, inlet for inoculation pipe, inlet for addition for acids or bases for controlling the pH. It consists of a stirrer to mixing of the fermentation media. Then it has a top plate, and the fermenter consists of a culture media. It consists of a pH probe and a temperature probe in order to detect pH and temperatures. It also has an oxygen sensor. It has a sampling point wherein we can collect the samples from the fermenter. It consists of a compressed air inlet so that necessary sterile air is sent into the fermentation media, and it also has a cooling jacket to control the pH, and it also has an harvesting pipe at the bottom of the fermenter. So how agitation and aeration is performed? Agitations are aimed to ensure similar distribution of microorganisms and nutrients in the broth. Aeration provides microorganisms with enough oxygen supply. The components required for aeration and agitations are like spargers, the aeration system, agitator also known as impellers, and baffles. Temperature control, adequate provision for control of temperature is necessary. Cooling jacket is provided to maintain the temperature of the fermenters. Pressure gas is used to measure the pressure of the fermenter. pH probes measures and monitors the pH of the medium. Inlet air filter provides a supply of air into the medium. Exhaust air helps in draining out CO2 from the medium. And control panel, it is a monitor which is used to control all the above parameters like temperature, pressure, pH, etc. Antifoams are necessary to reduce foam formation during fermentation. Natural antifoams include plant oils like soya, sunflower, and rapeseed. Deodorized fish oil, mineral oil, and tallow are also used as natural antifoaming agents, whereas synthetic antifoams are like silicone oil, polyalcohols, alkylated glycols, etc. Some of these may cause adverse effect in downstream processing steps, especially membrane filtrations. Next, moving on to types of fermenters. Industrially, three important types of fermenters are being used like stirrer tank reactor, bubble column reactor, airlift reactor. Stirrer tank fermenter or bioreactor, it is a small vessel made up of glass, while large vessels are made up of stainless steel of 1 to 1000 or even 8000 liters of capacity. They are closed systems in which volumes are fixed. They are agitated with motor driven stirrers. It is a kind of reactor with continuous flow culture system, which is either of chemostat or turbidostat type. Advantages of stirrer tanked fermenters or bioreactors is it is used for continuous operation or continuous fermentation. It has a good temperature control system, easily adapts to two-phase runs. It has a good control, simplicity of construction, low operating cost is there, and it is very easy to clean the unit. Disadvantages is consumption of more power due to the presence of mechanical pumps. It needs shaft seals and bearings. Size limitation by motor size, shaft length and weight. Applications: stirrer tanker fermenters are used in wastewater treatment, production of primary metabolites, single cell protein, lactic acid and methanol productions. This is the skeleton or structure of stirrer tanker fermenter. Here the main thing is it consists of baffle plates and sparges for stirring the media, and thus the name applied. And it also consists of sterile exhaust gas outlet, sterile filter, stirrer shaft seal, additional portion, then water or steam inlet, then outlet jacket, disc turbine impellers and stirrer shaft. Moving on to the second type of fermenter, airlift fermenters. It comprises of a gaslight baffled riser tube or draught tube which is connected to the downcomer tube. These airlift fermenters are basically of three types, like internal loop airlift bioreactors, split cylinder device, external loop airlift bioreactors. These types of fermenters are commonly employed for aerobic bioprocessing technology, wherein we grow aerobic type of microorganisms. Because of their high efficiency, it is mostly preferred for production of methanol and single cell protein production. Structure of airlift fermenter. Here we can see a draught tube inside the fermenter, and you can see a sterile exhaust gas outlet and sterile air inlet. As told in the previous slides, these airlift fermenters are of three types: internal loop airlift bioreactor, split cylinder device and external loop airlift bioreactor. The diagram A shows internal loop airlift bioreactor, wherein we can see a downcomer on either side, whereas split cylinder device you can see downcomer to one side. External loop airlift bioreactor, where the loop is present outside, and we can see downcomer and riser. Advantages of airlift bioreactors is it is very simple. The design is very simple with no moving parts or agitator for less maintenance, less risk of defects. It is easy to sterilization, low energy requirement when compared to stirrer tanker fermenter. It does not need energy for moving parts. Like in stirrer tank fermenter, we have seen the agitator shaft which is to be moved while in operation, whereas here there is no necessity of movement because of the presence of this internal loop or cylinder, as the device consists of a draught tube. There is no necessity for any shaft, and the cost is also very low comparatively. At the airlift bioreactor, it doesn't need the heat plate to control the temperature, because the draught tube which is inside the bioreactor can be designed to serve as internal heat exchanger. It is different to the stirrer tanked bioreactor that needs the heat coat or plate surrounding the tank to make warm bioreactor. It is clear enough that airlift bioreactor has greater heat removal capacity when compared to stirrer tanked fermenter. Disadvantages of airlift fermenter is greater air throughput and higher pressures are needed. The agitation on the airlift bioreactor is controlled by the supply air. To adjust the supply air, the higher pressure is needed, and if the higher pressure of the air needed, then more energy consumption is needed and more cost may be has to be paid. Inefficient inefficient break of the foam when foaming occurs, because there is no blades or shaft, as the foam cannot break when compared to stirrer tanked fermenters. No bubble breakers, just like stirred tank fermenters, which consist of sparges. Here they do not have any sparges, so bubbles which are formed during fermentation cannot be broken. Applications: it is used for production of mammalian cell cultures, wastewater treatment, biological processes involving biocatalysts as solids to produce biopharm proteins from fragile cells. Next, moving on to the third type of fermenter, bubble column fermenter. It is a cylinder with an aseptic height, aspected height to diameter ratio of 4 to 6 or 4 is to 6. The gas is sparged at the base of the column through perforated pipes, perforated plates, sintered glass or metal microporous sparges. Performance factors such as oxygen transfer, mixing, etc., are influenced by the gas flow rate and rheological properties of the fluid. Internal devices such as horizontal perforated plates, vertical baffles, and corrugated sheets packing may be placed in the vessel to improve mass transfer and modify basic design. The column diameter does not affect its behavior so long as the diameter exceeds zero point 0.1 m. One exception is the axial mixing performance. For a given gas flow rate, the mixing improves with increasing vessel diameter. Mass and heat transfer and the prevailing shear rate increases as gas flow rate is increased. Diagram of bubble column reactor, wherein you can see gas outlet. It consists of a CO2 gas cylinder which is pumped into the media in order to produce the bubbles in the fermenter, and it consists of sparging, then pH meter, then vacuum pump and temperature sensor along with a drain at the bottom of the fermenter. Advantages is simple design. It has good heat and mass transfer rate. Disadvantages are the sparger gets clogged because it consists of small holes. It gets clogged with the cells in the long run. Applications: it is used in production of baker's yeast, beer, vinegar, and in aeration and treatment of wastewater treatment. Thank you.